Hello once again, my name is Bombazimala Placid. So today we are going to talk about the immunology, specifically about the applied immunology. So I'm the student from the Ines Ruhengeri in the biomedical laboratory sciences in the year 3. So today we are going to talk about the immunology. Specifically here we are having the table of content or the objective which highlights what we are going to talk about in these few minutes. So stay tuned, be with us in this short period of time. So by the end of this presentation, we should be able to demonstrate the principles behind agglutination reaction. So those reactions which involve the clamping or the aggregation between the antibodies and the antigens. So the second point, we talk about how the latex agglutination work in the diagnostics. Means that we are going to look about the how the latex facilitate the agglutination so that it may help in the diagnosis. The third one is the principle of the precipitation reaction in the immunology. So the last one we talk about the radio immunodiffusion test. So how the radio immunodiffusion test is performed. So the first point is the what is the principle behind agglutination reaction. Specifically, the agglutination reaction, these are reactions which involve the antibodies and so the interaction between the antibodies and their particulate antigens. So which result in the visible clamping or the agglutination. So here they are said agglutination reaction are based on the clamping of the particles when antigen on the surface of the cell or particles interact with the specific antibodies. So here they are giving an example of the the blood typing. So in the blood typing, we are having the red blood cells. So the red blood cells, they are having the antigens on their surfaces. Means that these antigens incorporated to their red blood cell surfaces are the one to determine the type of a blood somebody is having. So in the blood typing, so we mix the red blood cells with the antibodies. So when the antibodies correspond to the specific antigens on the blood surfaces, especially red blood cell, their result or in their interaction result in the visible clamping. Their interaction are visible, are seen with the visible clamping. So this is one of the example behind the agglutination reaction. So here also we are having the figure which illustrate or explain how the agglutination takes place. So here we are having the latex which is coated with the antigens. So these antigen coated to the latex react with the antibodies. So these are antibodies which react with the antigen coated to the latex. So as a result, there is a reaction between the antibodies and the antigen coated on the latex. So this reaction or interaction, they result in the clamping or the visible clamping which is seen with the naked eyes. So means that on this point, so the agglutination, the principle for the agglutination lies in the interaction between the antibodies and their particulate antigens. So the second point is the how the latex agglutination work in the diagnostics. Firstly, the latex, these are the natural or synthetic polymers, especially derived from the polyester. So these act as the carrier which facilitate to hold either antigen or antibodies. So when it is used, especially in the diagnosis, it may it may be coated with a specific antibodies. Especially, let's have an example when they want to diagnose is like the Streptococcus aureus. So if this latex is coated with the antibodies against Streptococcus aureus, so if they took the sample and they mix with the antibodies coated within the latex and result in agglutination, means that they it indicated that in the sample they are testing, it correspond or it contain the corresponding antigen to the coated antibodies. So here they are said the latex agglutination test, it is a test done in the lab to check for a certain antibodies. As I have said, it may coat with a certain antibodies or antigen, and within the sample we are testing, either saliva, urine, CSF, or blood, if the corresponding antigen or antibodies is observed, it will be indicated by the clamping or the agglutination. So the latex agglutination involves the use of the latex beads, as I have said, with antibodies or antigen. When mixed with the sample containing the complementary antigen, so the antibodies agglutination occur, forming the visible clamp, as I have said. So here we are having the steps how this test is performed in the lab. So the first step is latex beads are coated with known antibodies or antigen. Means that you are having the latex beads. So these latex beads, it is coated with either antibody or antigen depending on what you are looking for. The second step is the introduction or addition of the sample. So after coating either antibodies, you introduce your sample to the latex beads. So the third one is if the target antigen or antibodies is present in the sample, the beads agglutinate, forming the visible clamp. So as the summary, the sample is sent to the lab. Maybe so it can be collected or either urine, CSF, or blood, and is sent to the lab where it is mixed with the latex beads coated with specific antibodies or antigen. So if the suspect substance is present, the latex beads will be clamped together or agglutinate together. So this test of latex agglutination results takes place either within the range of 15 to an hour. So if it perform this test, you may wait to get a results in the internal of the 15 to an hour duration. So here we are having the figure which illustrates this test. So here we are having this is the latex beads, so which is the form of the cycle. This is the latex beads coated with the specific antibodies. So this is antibodies coated to these latex beads. So this is all the latex beads coated with the specific antibodies. So here they are said there is an introduction of the analyte. So the analyte talking about here, it is the sample. So the sample may contain a specific antigen. So they are called it as the analyte, which represents the antigens from the sample. So here we are having the reaction between the antigen or the analyte, which is represented by which represent the antigen. So these antigen react specifically to the antibodies react to the antibodies coated by the latex beads. So these latex beads, as I have said, it acts as the carrier which hold the antibodies or antigens. So for this case, we are having the antibodies coated to the latex beads and the sample, which here referred to the analyte, which contain the antigen, they mix with or they react or interact with these antibodies coated to the latex birds. So they result or they result in the formation of the close link and agglutination so the agglutination indicated that there is a close link or there is a formation of the bond or interaction between the antigen and the antibodies. So the next point is the what is the principle of the precipitation reaction in the immunology. So the precipitation reaction, so these are reaction which takes place between the antibodies and the soluble antigen in the aqueous solution. So here they are said precipitation reaction are those reactions in which antibodies and the soluble antigens interact in the aqueous solution to form the lattice that eventually develop into the visible precipitation. So the lattice they are saying here, so this lattice, it is the a net-like structure. So a net, so these are which have the similar like these prevent the interest of the mosquito or those used in the catching the fish. So this lattice means that there is interaction or interconnection or the cross linking formation between the antibodies and the antigens. So this lattice formation to be formed, it requires the complementarity between the antibodies and the antigens. So here they are said the principle for this the precipitation reaction, they are having the antigen which is soluble react with or interact with the antibodies, resulting in the formation of the antigen antibody complex which is insoluble, or the precipitation or the lattice. So the reaction which takes place between the soluble antigens and the antibodies, they result in the formation of the insoluble precipitate or the lattice. So as I have mentioned above, so this interaction or the formation of the precipitate, they need or they depend on the variance of both antibodies and the antigen. So the antibodies must be bivalent. A precipitate will not form with monovalent fragment antigen bindings fragments, means that the antibodies, they must have more than one epitope to bind with. Then the antigen must be bivalent or polyvalent, means that having ability to bind more than one antibodies. That is it must have at least two copies of the same epitope or the bindings of the antibodies to the antigen. So these antigen, they must possess more than two copies of the epitope, means that it must have ability to bind or to be binded by more than two antibodies. So different epitope that react with different antibodies present in the polyclonal cell. So the proportion of the antigen and antibodies in the reaction must be equivalent for the precipitation reaction to occur. So means that for this reaction to take place, they need the complementarity between the antigen and antibodies. Once there is an imbalance between the antigen and antibodies, they result in the formation of the what you call post zone, pro zone. So the post zone, this is where we are having an excess antigens in the solution, while the pro zone, when we are having the excess of the antibodies in the solution. So here they said that in case of the imbalance of the between the antibodies, that in the zone of the equivalence, meaning that so here for the zone of the equivalence means that the optimum antigen antibody ratio leading to the maximum precipitation, means that if there is an equivalent between antigen and antibodies, they will result in the maximum precipitation. But once we are having more or excess antibodies, there will be no precipitation due to the insufficient antigen bindings. So you can imagine how the excess of the antibodies will result in no precipitation. So as you have seen that the antibodies, in case of the excess antibodies or higher concentration of the antibodies, means that the binding site of the antibody to the antigen will be in higher concentration, means that the antibodies will be in higher concentration and they will exceed their bindings to the antigen. Once they exceed their binding to the antigen, means that the antigen will bind in a single form to the antibodies. Once they bind in a single form to the antibodies, means that there will be no formation of the cross link. Once the cross link is not formed, this will result in no formation of the precipitation, because the precipitation will form when there is a cross link, meaning, or in other words, when there is a multiple reaction of the antigen, or when antigen bind in multiple way to the antibodies. Once there is a higher concentration of the antibodies, means that the antigen are enabled to bind times or twice or even more than two to the antibodies. But on the case of the antigen excess or post zone, on the same way, so there will be no formation of the precipitate because the antigen are insufficient, means that their bindings are saturated by the antibodies. Once the antibodies, once the antigen exceed the antibodies, means that their availability in the solution will be in higher concentration, means that there will be no formation of the cross link. Once the cross link is not formed, the precipitation also will not be formed. So here we are having the figure which illustrates the how the lattice is be formed, or the how the precipitation will be formed depending on the number of antibodies and the number of the antigen reacted. So here we are having the specific antibodies of interest coating insoluble particles such as polyethylene. So as I have said, so this is the latex braids which coated with the antibodies. So this latex, as I have said, it is driven from the polyester. It is also considered as a natural or synthetic polymers. So these latex braids which hold or coat the antibodies, they react with these antigen will react with the patient or the serum antibodies. So their interaction will lead to the formation of the formation of the complex. So these complex will be precipitated in the solution because there is the sufficient or optimum between the antibodies and the antigens. So here we have, you see that this is the lattice or the insoluble precipitate because there is saturation of the antigen and the antibodies, means that the antigen is able to bind to more than one antibodies, which result in the formation of the cross link, which is one of the most factor in the precipitation reaction. So here also we having another figure which illustrates how the excess or deficiency of the either antibodies or antigen will result in either formation of the precipitate or not formation. So here we are having the antibody excess zone. So I've said, so when we are having the more concentration of the antibodies, means that the antigen are unable to bind to all antibodies, or the binding site of the antibodies are low compared to their concentration, means that there will be no formation of the close link. Once the close link is not formed, so the precipitation will not be formed, or the formation of the insoluble precipitate will not be formed because of the imbalance between the antibodies and the antigen. So here we are having the equivalent zone, means that there is an equal concentration of the antigen and antibodies, means that the antibodies and antigen, they interact equally, means that the antigen is able to bind more than one antibodies, which result in the formation of the close link. But for this antigen excess, or what you call the post zone, it is where we are having more higher concentration of the antigen in the solution. So if we are having more concentration of the antigen, means that the formation of the close link is tightly low. If the close link is not formed, so there will be a higher chance of developing the soluble precipitate instead of insoluble precipitate, because of the incomplete or incompatibility. So the sorry between the there is an equal between the antibodies and the antigens. So for this reaction to take place, they need the complementarity of the antibodies and the antigen. So means that we need to pay attention on the quantitative, or in the measurements of either the antibodies and the antigen to have a sufficient reaction. So the next point is the how is the radioimmunodiffusion test is performed. So the immunodiffusion, these are technique which is used to measure either antibodies or antigen by their precipitation, which improve diffusion through the substance such as agar or gel agarose. Means that you are having either antibodies or antigen suspended in the agar or agarose, or in other medium. So to know the concentration of the either antibodies or antigen, you measure the ring formed as a result of interaction between antibodies and the antigen. So here they say the radioimmunodiffusion is a single diffusion technique whereby a solution containing the antigen is pressed into the well in a gel or agar surface, even when impregnated with the antibodies. So the diameter of the ring that precipitates around the well as a result of antibody reaction corresponds to the amount of the antigen in the solution. Means that there is the plate which contain agar or agarose with impregnated with the antibodies. So the sample introduced to that plate, it contains the antigens. So the ring formed as a result of interaction between the antigen and antibodies will be measured in the diameter. So the diameter of that ring will correspond to the concentration of the antigen in the sample will correspond to the diameter of the ring measured. So that is what it is explaining to this paragraph. So here we are having the procedures how this test is performed. So the first one is jelly preparation. A jelly contain a uniform concentration of the antibodies. Uh, this jelly contain a uniform concentration of the antibodies prepared on the plate. Means that we are having the plate which contain the gel, and that gel contain a concentration or a uniform concentration of the antibodies. The second procedure is the sample amplification, or means that there is the addition of the sample on that plate incoated by the antibodies. Then there is diffusion. It's the antigen diffuse largely, forming a ring of antigen antibody precipitate. Means that after adding the antigen to the or adding the sample to the plate containing the antibodies, there is interaction between the antibodies and the antigen. So this interaction will cause the ring. So this ring, it is one which based on to measure the diameter, and the diameter correspond to the or is proportional to the antigen concentration. So that is what is said. The measurements, the diameter of the precipitation ring is proportional to the antigen concentration. So here we are having the figure which illustrated clearly. So here we are having this is the plate. All of this is the plate, right? So this plate, it contains the antibodies incorporated into the agar. Means that this plate contains the agar, which is the media, right? So these media incorporated by the antibodies, or there is an impregnation of the antibody to the agar. After that, there is an addition of the antigen to the agar. If there is an addition of the antigen, there will be an interaction between the antigen and the antibodies. Once the antigen interact with the antibodies, there will be a formation of a precipitate. A precipitate means that the antigen will diffuse. To diffuse means that they will the formation of the antigen antibodies will result in the formation of the ring. So this ring indicating that there is an interaction between the antibodies and the antigen. So to know how much or at what extent our sample contain the antigen, or the at what percentage or at what concentration does our sample contain antigen, so we will measure the diameter. Means that from this point to this point, we measure the diameter from here up to here. So to note the distance or the diameter of this ring will be in proportion to the concentration of the antigen contained in the sample. Means that the diameter we measured on this ring, it will indicate how much antigen our sample was possessing or was having. So this is how the immunodiffusion test is performed. Means that you are having the plate which contain the agar or J agarose. So on that agarose, it is impregnated by the antibodies. So in that agar contain the antibodies, you introduce the antigen. So there is an interaction between the antigen and the antibodies, which is the formation of the ring, precipitating ring. So this ring, you measure the diameter of the ring to know the concentration of the antigen, as the diameter is proportional to the concentration of the antigen. Right. So finally we are having the reference. So where we took the information we were discussing. So here we use the textbook which called the Kubi Immunology by Jenny Put, Sharon Stafford, Patricia Jones, and Judy A. So if you need more information about to this topic you are discussing at, so you may contact to this book for more details. So thank you so much for your kind attention, for listening to us. Thank you.